So my computer doesn't. There we go. Okay, so as a reminder to those of you who may not have joined before, so these journal clubs are about ultimately trying to get towards the Aspirational Neurosciences Memory Decoding Challenge, where that is, we're trying to encourage people to demonstrate decoding of a non-trivial memory or some other learned function based from only the static map of synaptic connectivity. So think of an animal brain, ex vivo, post-mortem. You're examining the synapses and you're trying to say, this zebra finch sung this particular song. We're able to work out uniquely what it was singing. Or we're able to say, this mouse ran through this particular route this way. It was able to navigate, and we're able to pull out the locations where it was, or any of these sorts of things from a static connector map. So the previous set of journal clubs we've been looking at have been mostly focusing on sort of examples of experiments that we think would actually win this memory decoding challenge. So something like, for example, being able to look at the zebrafinch brain and pull out the song that zebrafinch sung. And we're going to continue to look at these sorts of things going forwards. But this week's a little bit different. We're going back to our usual journal clubs, which we were doing last year, which instead of focusing on what's the sort of experiment that could win the big prize, we're just focusing on papers that are sort of incremental milestones towards winning the big prize. So each year we give out awards, annual research awards for papers that have developed a new technique that we think will be really useful for decoding memory, or that have made some other sort of scientific incremental milestone along the way. And the paper that I'm discussing today was one that was nominated late last year for a potential annual research award. So we're going to talk through it, see what's in it. And it will be a nominee for this year, 2026's, at least it will be one of the papers that's considered as a nominee for this year's awards that will be coming up late this year at SFN. So we have Engram cell connectivity as a mechanism for information encoding and memory function by Ortega-de San Luis et al. And this comes out of the Ryan lab in Dublin. So Thomas Ryan has done a lot of this his lab's done a lot of work on engrams and sort of behavioral experiments. He also, so this paper focuses a little bit on the molecular mechanisms that underlie sort of engram cell connectivity, which makes sense because my memory is that is his PhD background was with Seth Grant in, I think somewhere in Scotland, that was looking a lot at like synapse formation before he did his postdoc with the Tonegawa lab, the famous engram lab at MIT. So it's sort of putting all of those bits together to a certain degree, and we'll see what's in this paper. So let's jump into it. So it starts, as these often do, with some sort of complicated behavioral setup. So they're using contextual fear conditioning here. So their readout from any sort of experiment is going to be, they take an animal, they put it in a particular environment, whether that's context A, context B, context C, as you can see marked in red or blue or orange here. And I'll talk through this in a second, so you don't have to understand all of it straight away. But just know that A, B, and C are different contexts. So they have different shapes, they smell different, they'll be noticeably different for the mouse. And what specifically differs between these is just a couple of the different steps in these different paradigms. So let's run through linked up the top first. So what they're doing initially is they're putting the animals in this orange context C. Then they're habituating them to this environment A, which just means they take them and they put them in there for, I don't know, half an hour or something, an hour. I can't remember exactly how long. Then 2 days later on day 4, they do the actual contextual fear conditioning. So they put them into environment B, different from A, and they do a series of 4 shocks while they're in that environment. Now the mice are scared of B. Then a few days later, they put them back into A, which they'd previously been in, where they weren't shocked, and they give them a single foot shock. And then the next day, they put them into back into A, which they now label as A prime, because while it's the same environment as A, they've since gotten a shock, which was different from the original A. So it's A prime. And they see how much the animals freeze, because mice freeze when they're scared of an environment. And then the next day, they check them out in context C, this neutral context, which is the one they started in. And the next day, they put them back into context B, the one where they were properly scared, to check that they're actually really quite scared of that environment. So that's the linked paradigm. Then they have a couple of not linked paradigms. So in the second one, not linked 1, what they do is instead of initially habituating them to A, they just leave them in their home cage on day 2 and then go through everything the same. And then in this not linked 2, this other paradigm, it differs in that when they put them into A on day 7, they don't shock them. They just it's A the same as it was before back on day 2. And what they're trying to work out through this is what sort of properties of the environment are needed to potentially link their painful experience in context B with their experience in context A. Yeah, that's just me describing, pulling out of the text what exactly they did in A. So what you can see here is that in context C, initially, they don't freeze at all. They're perfectly comfortable with C, which is good. And in context A, initially, they don't freeze at all, which is also good. And you can see at the end here, in context B, they're all freezing substantially, like 60% of the time. So they all do get scared of B. And then the question is, what's happening in A prime? And you can see for the animals in the not linked 1 or the not linked 2 paradigm, they're not freezing when they're placed in A prime on day 8. That makes definitive sense for the not linked 2 because they never got shocked in A. But it is interesting for the not linked 1 that they don't freeze in A prime because they did get 1 foot shock while they were in A prime on day 7. But that is not enough for them to start freezing substantially. It's much less than for those that were habituated to A on day 2 previously. Yeah, Randall, go for it.
Yeah, I was just wondering. So in the not linked 1, so I know it's only a single foot shock, so it's not a very long experience in which to learn. But the exposure to the environment, is that also very brief? Or is that of some specific longer duration as well?
That's a good question. I don't know. We can check the methods quickly to see if they changed how long they left them in there for. Let's see. Behavior, context A, timelines, free to explore. For the updating, mice were introduced to the arena. They received a single foot shock. They were removed from the arena 1 minute after.
Pretty brief.
In the control group that did not receive an update, mice were allowed in the arena for 1 minute. No foot shock was delivered. Yeah, so it's only 1 minute for any of them.
Mm-hmm. Got it. Thanks.
That is fast. That's a good point. I didn't pick up on that before then. So yeah, they need to have been exposed for longer previously to be able to form the memory, the linking memory on day 7. Where was I? Yes, the mice start to only the ones in linked start to freeze substantially. So ones that had been habituated to A previously, so they were able to have a sense of what environment A was, rather than just being thrown in for 1 minute with no context, having their foot shocked, and then pulled out again. So this is the starting behavioral paradigm they're going to use throughout the set of experiments that come next. Okay, so, yep, Ken, you're muted. Ken, you're muted. I can't hear what you're saying.
Sorry, on the previous slide, is there a human language explanation that we're supposed to be thinking of for why the not linked is different from the linked? in this case, because it's kind of like, and maybe you're going to get to this, but you know, from the mouse's point of view, it was shocked in both, and you know, and let me naively, it has more evidence in the linked that A is not dangerous. In the not linked, it's only seen A once, and it got shocked. So, you know, you would think it would be hell. A is bad, and it's always bad. Maybe I'm anthropomorphizing this, but at this point, are we supposed to be thinking?
You're only talking about linked one, right? When you're comparing linked to.
That's right.
Linked.
Only linked one.
Okay.
Yes.
Yeah, no, I agree. I was also confused and had to read this a few times, and I'm still, having finished the paper, unclear on why it works that way. I mean, empirically, like, that's what their behavioral results show, and that's why it's figure 1 inlet B, because I agree, it's a strange... You could imagine these results going the other way around, but that just, like, is what they've shown here. So I don't have a human level.
This was actually the reason why I asked about the duration that they were exposed to A, because in not linked, if the A exposure is really short, maybe a minute isn't enough to even get to know and be able to recognize the context.
Yeah, this is what I was thinking, that it's like they don't really understand that this is a unique environment that they can recognize when they get tested again, that they're in the same environment.
Unless they've been in there for a while previously.
Yeah, because I assume the habituation phase is longer.
Yeah, I think it was half an hour at least. Half an hour. Yeah, no, sorry, Ken. It's just empirically rather than through some clear a priori reason, at least to me. So that's their initial behavioral protocol. Then they also do some labeling of what's going on. So they are taking recordings from the basolateral amygdala, as we've covered multiple times. They use an RFP label that is under the control of CFOS and docs. So the idea being that they take a group of animals, they run them through the linked and the not linked protocols. For day 4 on training, slightly before and during, they take them off the docs so that any cells in their basolateral amygdala that are active will be displaying this RFP after they have been labeled. And then they run them through the paradigm as we've seen previously, and they look at the labeling in the basolateral amygdala. So you can see here some of the images that they take, which, you know, looks like there's RFP expressing cells there. And then what they show with their actual histology results is that if you look at the proportion of cells RFP of DAPI, so that basically means how many cells, so DAPI is just a cell marker generally, it lights up blue, and RFP is cells that were active during the experience in B, so during the scary experience in B. So basically in D, what you're seeing is what proportion of basolateral amygdala cells were active during scary experience. You see around 7% in both groups. In E, we're asking what proportion of cells had this immediate early gene activation, CFOS, as part of the basolateral amygdala on day 8, rather than on day 4, which is what D shows. So this is when they're put into A prime again, just before they're sacrificed. And you can see, again, they have very similar levels of response. But what differs is this F1, which is what proportion of overlap is there between those populations. And you can see that the overlap proportion is higher in those that were linked versus unlinked. So this is their argument that what is happening in the linked scenario is that more of these cells that were active in B now also become active for A in the basolateral amygdala. So they've linked the scary experience with B with the previously neutral experience of A due to day 7 sort of linkage experience together. And this is sort of the key argument that they take forwards through the rest of their sets of experiments. Does that make sense to everyone before I go on? Cool. All right. So keep that in mind. So then they start to play around with the circuitry to see if they can understand it a little bit better. So they're asking, in this circumstance, okay, what happens if we've got the cells in the basolateral amygdala labeled using our doc system that I just ran through? What if we also label the cells that were active when the animals are initially placed into A and habituated to that context, and we label the cells in the CA1? So within the hippocampus that we think are marking the cells sort of contextual memory. But this is the area that's typically used in a sort of contextual Engram style experiments. Whereas the BA is typically more associated with the animal's valence experience. So they're like, if we label these cells that mark the context, hopefully of A versus B versus C, in ventral CA1, will that tell us anything? And particularly, if we reactivate those cells in a neutral context, in neutral context C, what is that going to do? How will the animals respond when they have the cells activated that make them recall context A? Will they respond neutrally? Will they respond by being scared? So they're using this channel rhodopsin 2 sort of linked to CRE and 4-OHT marker that was introduced when the animals were placed in A on day 2. And then on day 8, after they've had the memories linked or not linked, they're going to activate it and see whether that causes the animals to freeze. And you can see from their behavioral results that you do get a somewhat of an effect in terms of increasing freezing in the animals that have been linked versus the animals that have had it not linked, implying that even just reactivating the cells that mark the engram for experience A after a linking experience is enough to induce this sort of fear memory in the mice. But I note that it's a bit of a weaker effect than the actual behavioral experiments, purely behavior experiments that we saw previously. So this is just to establish that, it's the cells that were active during A that are important for this memory recall later. So then they also combine that with labeling B while also reactivating A at the same time. Labeling B, reactivating A at the same time. So now we're looking at the basolateral amygdala again. And they're essentially replicating the results that they had previously, where if you look at what happens in the basolateral amygdala after you've been artificially reactivating their experience of being in A, if you look at the number of cells that are active in the BA, it's the same. If you look at the number of cells, the number of cells that were active during the scary experience on day 4, it's the same. If you look at the number of cells that were active just before the animals were sacrificed on day 8, it's the same. But if you look at the overlap, you see that there's much higher overlap in the animals that had the linking experience versus the animals that didn't have the linking experience on day 7. Okay, so that's establishing, again, through this optogenetic means that this linkage is actually occurring as a result of their behavioral experience. That's in a sort of positive extent with using reactivation of the cells that were labeled on day 2 in context A. How about inhibition? Can they knock the fear memory out as well? So this time, what they're going to do is they are going to label 2 different sets of cell populations. Either they're going to label the cells that were active on day 4 when the animals were in their scary experience in context B, or they're going to label just a random set of cells in the basolateral amygdala that were active when the animals are in their home cage. So that's what they call random ensemble. And what they're going to do is on day 8, they're going to inhibit either the cells that were active on day 4, or they're going to inhibit a group of cells that were randomly active at some random time. And they're going to do this in a group of mice that have gone through the linking experience. So these mice have all, they've had their habituation to A, they've had their scary experience in B, they've had their linking experience to take it from A to A prime on day 7. So all these mice should be freezing in response to being placed back into A, but they're going to see what happens if they inhibit this population of cells that was active during B, or a control random population of cells. And indeed, the results go in line with what you would expect, which is that inhibiting the cells that were active during B, even while the animals are placed in A, does reduce their freezing. So this implies that the scary experience of A and the fact that they're now freezing in A does have something to do with the experience that they had during B. And if you turn off those cells that were active during B, then you reduce the amount of freezing overall. Now, I just want to pause for 1 second. I'm going to come back to this in the discussion, but I am a little confused as to whether they are implying by doing this that they have inhibited the memory of B as in the context of B, or whether they've just picked a population of cells in the BA that's important for experiencing negative valence, and they've essentially just like toned down the animal's ability to experience scary experiences overall. So that's just something to keep in mind as we go forwards. Randall.
Sorry, maybe I just didn't hear that correctly, but wasn't that exactly the comparison they're trying to do there between the two? Because one is a random set of cells in BA and the other one is a set of cells that were actually involved with stimulus.
Yeah, but there's subpopulations of cells within BA that control different sorts of experiences, or different levels of valence. My understanding, I mean, I need to be a little bit more on top of BA, but I guess my question is whether they have, just in the first context, in Engram B manipulation, whether they've just been able to specifically target the cells that are controlling negative valence, whereas if you do a random ensemble, I guess another way of putting it is if they had done this experiment while placing the animals back into any scary experience they'd had, whether it was linked to B or not, would this inhibition have toned down their freezing response? Or is it specific to things that are linked to context B, is what I'm unclear on.
I see that. Okay. Yeah, I guess I'm still not getting it, because I thought that by targeting on day 4 here in training, they're labeling on that day and the top row. You would assume that this is then during that experience. So it is that particular fear experience, the experience of that foot shock, and whatever valence is being coded for, which we assume is a negative valence, that's what they're then tagging. That was my assumption.
Yeah, that's the assumption that I think that's a reasonable assumption, and the assumption that they the paper goes with. But I'm just curious, because it's BA instead of, let's say, the hippocampus, whether they have picked something that is specific for negative experience in context B, or whether it's just negative experience generally. And then if you imagine a different paradigm where they also gave the mice a scary experience in context beta that was unlinked to anything else, and then they put them back into beta later, and you did this inhibition of those particular cells in BA, whether the animals would be reduced in their fear, or this population would have nothing to do with that memory.
Yeah, I think I understand what you're asking now. Yep.
Yeah. Other people listening might have a better understanding of the BA. I know we've covered it a few times, so you might be able to tell me why I'm correct or incorrect, but that's just a thought that occurred to me as we were going on. Okay. So yes, that's what they're demonstrating with this one, that the negative experience in A requires the cells that were active in B, and that inhibiting those cells that were active in B on day 4 reduces the animal's ability to freeze when placed into B on day 8. Okay, so that's most of the behavioral work that they've established and the engram cell level. Now what they wanted to do is to look at, what are the actual synaptic molecular mechanisms that are helping to encode these changes that happen during the initial memory formation and the changes specifically that happen during the linking experience that links context B to context A with that updating event on day 7. So in order to do that, they say they analyze dendritic spines and synaptic markers and morphology, and my impression is they just analyzed a bunch of stuff, and some of what they analyzed made it into the paper. So the first thing they look at is just sort of gross morphology in terms of number of spines, spine volume, and average spine head diameter. And they look at this in animals that had the linking or didn't have the linking experience, and that go through a recall experience on the last day, day 8, before they're sacrificed, or that don't go through a recall experience. And they show that almost nothing changes between the linked animals or the unlinked animals, and those that were subject to recall or no recall, except for spine density, where they show that the spine density in the basolateral amygdala goes up for animals that were subject to the linking experience and subject to the recall test. I'm a little confused on how to interpret this, but that is what they say happens. It also doesn't appear to be a huge effect. Yes, Ken.
Sorry, it might be written right here, but are these specific cells that were labeled in BA?
These were also a good question. I think hold on. I'm trying to pull it up. Were they the RFP cells, or were they? After recruiting Engram B cells, yeah, I think they are the RFP labeled cells. So the B cells. After recruiting, we analyzed the battery parameters in the Engram B cells. At least they say it's the Engram B cells.
Okay.
So the cells they know are involved in that learning experience for B. Yeah, okay, I guess this helps clarify this as well. So they're looking at these RFP labeled cells against the background of DAPI cells. The RFP is labeled in pink, which I guess also helps them actually look at the dendrites and the spines because they're glowing. And I know they used a confocal microscope with fluorescent to examine these things. So I presume that would also help with their imaging here. And this is them looking at excitatory cells for either the linked or the not linked cohorts. And they're looking at RFP, which just labels the cells, the Engram cells. And then they look at a number of other markers. So they look at PSD95, synaptic post-density marker. They look at DAPI, which is just for the cell bodies. And they look at VGLUT1, which is temporarily escaped me what VGLUT1 is. I presume a glutamate receptor, but yes, this is what they're examining. And again, they mostly find negative
presynaptic marker.
Yeah, that's right.
Glutamate transporter.
That's right. Yeah, it's for vesicle. Yes, correct. Thank you. It's presynaptic. So VGLUT1's their presynaptic marker. PSD95's their postsynaptic marker. And they show mostly, again, no effects, except for they see decreased PSD95 expression in the Engram cells that were in the linked experience relative to the not linked experience. And this brings us back to what I was talking about with Randall at the start of the call, which is the question of what is happening with PSD95 in these animals during these experiments. And we will be coming back to that in 2 slides. But just flag for now that what they're seeing is in the animals that are undergoing linking, they see decreased PSD95. They also do the same sort of imaging results in animals that, from the inhibitory cells instead of just the excitatory cells. So this time they're looking at GABA markers, essentially, and they show that there is decreased GPHN puncta, so inhibitory puncta, onto the engram cells from animals that have undergone the linking, as opposed to the unlinked groups. So the 2 changes they're seeing are decreased inhibitory puncta and decreased PSD95 in these engram B neurons in the BA. But nothing else seems to change that they've looked at. So not super dramatic molecular changes. All right, so to come back to PSD95, they spend the rest of their experimental time sort of focusing largely on PSD95. So here's a little schematic of a synapse in the top left. And for those of you who maybe haven't seen these things before, basically what they're showing off is on the cell membrane, you have all these actual receptors, your NMDA receptors, your AMPA receptors, but anchoring these things into the synapse is a whole bunch of postsynaptic density proteins, of which one of the most important ones is PSD95, labeled here this like long, thin green thing. And the presence or absence of PSD95 is associated with, the size of synapses and the formation of synapses. And given that they've seen these changes in PSD95 that occur during linking, they were presumably curious about what is the role of PSD95 during the linking experiences in these animals. So what they decide to do is they decide to, in these animals, to knock down or leave the PSD95. So they use, they have a setup where they have the mice that if they remove docs or they expose them to docs, they will either produce or not produce PSD95 that is also RFP labeled. So they can actually observe whether they have knocked down or they have left PSD95. And you can see an image of that on the bottom left in these PSD95 knockdown or sort of scrambled random control animals. I will say as well, I found confusing what exactly they meant by scramble in the paper. They could have more clearly defined it. I'm pretty sure scramble means virus that didn't have this DLG4 knockdown, these short these RNAs to knock it down, but I'm not entirely clear. Randall?
Yeah, no, sorry. I agree with you on the scramble thing not being super clear. I'm taking you back to the image that you're showing us of what PSD95 is doing there in anchoring the receptors. And I'm wondering in this image, because this is an image that isn't in the paper, right? This is an image you got somewhere else, I think. Yeah.
Correct.
The colors of those receptors up there, do they mean something? Is one of them NMDA and the other is AMPA? And is PSD95 only involved with anchoring NMDA or something like that? Or
this is like going back on my memory. I don't. I feel like I want to say that it is more involved with anchoring NMDA receptors, but I am absolutely not confident that that is true, and it's not something I've looked into for 7 or 8 years or something.
Yeah. Now I'm probably going to go try to look it up because I'm curious if that explains some of the confusion I've been having about PSD95, because you could, for example, make a story where if PSD95 is more involved with anchoring NMDA and you see a reduction in PSD95, that it changes the proportion of NMDA to AMPA receptors, something like that.
Yeah,
maybe.
That's a good question. So they've developed this way of knocking down PSD95 levels in animals at specific time points. So what they use that for is they take animals that have gone through the linking paradigm, and on day 4, they knock down PSD95 formation in the synapses that are formed during their scary experience in context B. And then they see what happens as a result of this. They are asking what happens to the animals when they experience A, context A, after the linking, if you've knocked down the level of PSD95 that is being formed on day 4 in scary experience B. And what is interesting, as you see here, is it doesn't seem to affect their memory in any way for the experience when you put them into hold on, why is that target CX test? Target context test. Oh, because they have different, sorry, just getting confused because they use C in a few different ways. But yes, if you put them into A prime after the linking experience, both the standard mice freeze, but also the PSD95 knockdown mice freeze. So it doesn't prevent the linking experience from occurring. If you look at the number of cells that are active in the BA, as shown in E, you see that there's no changes. What you do see changes to is the number of active cells in context A and the level of overlap between cells that are active in context A and cells that were active in context B. So what seems to be happening in these PSD95 knockdown animals is an increase essentially in the number of these cells that are active, which, as per what I was discussing with Randall before, seems a bit strange to me from my previous understanding, in that it's showing that by knocking down PSD95, you're getting an increase in excitation, which was just new to me when I was reading the paper, because I had previously thought about PSD95 as something that stabilizes synapses, enhances synapse formation, those sorts of things. Okay, I didn't see a hand, but I will then continue. So they take this a little bit further, and they take the previous behavioral paradigm that I just mentioned, and they say, okay, if it seems to be the case that there's no differences in the knockdown and the standard animal's ability to remember context A after linking, and we're seeing, in fact, increased excitation as a result of removing PSD95, does that mean actually maybe the memory is stronger as a result of this? The linking has happened to a stronger degree? So what they do is they then place the animals into these various different contexts to test their memory, whether they'll freeze in a neutral context, because maybe they're overgeneralized and they're just freezing all the time. They see what happens when they put them back into context B. And then they also see what happens during an extinction paradigm, when they try and take the animals and they see whether they're able to unlearn their scary memory if the negative stimulus is taken away. And what they find is that the animals still, they have no difference in the degree to which they freeze when placed in a neutral context. So they're not massively overgeneralizing. And they have no difference in the amount they're freezing when placed into context B. So they're not just like generally freezing more once PSD95 is knocked down. What they do see is a difference in their extinction levels. So they show that by knocking down PSD95, these animals are, they hold on to this memory of context A being scary more strongly than animals that had intact levels of PSD95. So then they take this, and for their last set of behavioral experiments, I think before we get to the discussion section, they this time see, well, what happens if we, instead of running them through purely this behavioral setup, we also artificially reactivate the neurons in CA1 that monosynaptically project to BA. I should have mentioned that up front, sorry. The whole basis of this work they're doing is that there's this direct monosynaptic connection between the CA1 and the BA. So this time they're doing the same linking paradigm, but then instead of putting them back into A directly, they're reactivating neurons that were labeled during A on day 2, but while the animals are in a neutral context C. And what they show as a result of this is essentially that the animals that had the knockdown make as previously seen, a stronger memory, a stronger linking between context B and context A. And again, the same sort of results where there is a strengthening of the level of activity and the reactivation in the animals that had PSD5 knockdown. Okay, so that brings us to the end of the results. Does anyone have any more questions before we get into what this actually means?
I'd say let's get to the end and then talk about it.
Yeah.
I checked up what PSD95 is doing for AMPA and MDA. Just asked Gemini, and it did not make things more clear. It made it less clear for me.
Okay. All right. Well, so to just quickly go through this, what this study does show is that you can link these previously independent memories through changes in synaptic wiring between engram cells. So they show that the output of these VCA1 engram cells onto these BA cells changes based on updating from A to A prime based on that single foot shock. They show that these engram B cells, they show increased dendritic spines, but decreased PSD95 and reduced inhibitory puncta after they've gone through this linking experience. And they show that this synaptic rewiring is favored by a reduction in PSD95. And indeed, artificially reducing PSD95 levels leads to even stronger linking. That, though, doesn't really give me enough to really understand the synaptic mechanisms that are going on here and what the role of these actual molecular changes at the engram cell level are doing. So there's quite a few things that I'm still confused by. As I mentioned before, I'm confused independently of that as to whether the cells that are labeled in B are an engram, so specific to negative valence in context B, or whether they're what I've heard Thomas Ryan himself call an engram, which is they're more a marker of instinctual negative experience. Are they just marking negative valence generally? I don't think they tease that apart in this particular paper. I'm also just very confused as to what PSD95 does. And maybe if I went away and read a lot more papers on the role of PSD95 in excitation and inhibition, I would come to understand that. But through reading the discussion of this paper, I am not there. And indeed, the authors themselves give several different theories that they're unclear on as to what's happening with it. And in particular, the thing that I'm confused by is the fact that there's increased dendritic spines in these engram cells, but that doesn't have anything to do with these PSD95 or any of the other synaptic markers that they had levels. So it's clear that the increase in PSD95 or decrease isn't affecting spine formation. The authors themselves note that E-grasp would have been quite useful in this set of experiments, where that's a technique that we have seen in many of the other papers that we've been covering, where you can specifically label the presynaptic and postsynaptic compartments of these cells, so you can distinguish between engram cells and non-engram cells in both CA1 and in BA, and the differences that happen in engram to engram cells, engram to non-engram cells, and so on. And indeed, would have been a better paper if they'd been able to put that in, but I'm sure that's very hard to do. So yeah, I essentially, in terms of takeaways for memory decoding, I agree that this paper does demonstrate that you can get this memory linking, which is interesting as a behavioral paradigm and not one I'd come across before. So that is a useful technique that you can imagine using in a sort of memory decoding behavioral experiment. But otherwise, I did not find it particularly helps me think about how I would design an experiment to perform a memory decoding sort of experiment. I did note that one of the things they show is that you can get these changes in molecular level. So you can have your PSD95 knockdowns or standard mice, and you can have these changes in wiring where they show, increase or decrease in the level of overlap in engram cells that are used to encode a particular behavior without really showing much change in the freezing that happens in the mice. And that's basically just a demonstration of redundancy, which might make things harder to distinguish in any particular sort of memory decoding experiment. But yeah, in the end, I think it's a cool paper, but not one that really helps me think about memory decoding too much better than when I started. But still a good paper to have nominated and to have read through. That's where I got to it. So now it's open. Anyone can talk from here. Random.
All right. Yeah, so I'm still confused. But so hi, I'm an alcoholic.
Yeah.
No. Yeah. So you mentioned that they have a number of different hypotheses, right? And one of them just kind of stood out to me, and it added to this whole notion of there being redundant mechanisms and it being really complicated to figure out exactly what's going on here, because if I didn't read it wrong, I think they also mentioned something like when they're trying to apply extinction, that it could be that this is adding a specific extinction engram rather than just removing the previous engram, so that there might be another thing being learned. If I read it correctly, I might not have read it correctly there.
So I can't remember the exact idea. Are you saying?
Let me see if I can find that.
Yeah.
Yeah. There's a part in the discussion where they talk about the extinction deficit shown by the decreased in this study can be both interpreted as a suppression problem caused by an imbalance between the original fearful engram neurons that outnumber the extinction engram, or as a modification consequence of a higher number of connections that could be therefore more resistant to weakening. Yeah. So there's this idea of an extinction engram.
That's not that controversial though. I mean, extinction is a different process to, I guess, forgetting in a sense, where there's a lot of behavioral work that shows extinction requires.
That's exactly it. It's the distinction between, say, forgetting of an existing engram versus the recruitment of a new extinction engram. So yeah, then that comes in, and so then you can look at the effect of the PSD knockdown in two different ways. You could see it as the effect on the existing engrams that are coding for that linkage versus, say, the effect of having knocked those down on trying to create or recruit a new extinction engram. So the effect can go both ways. Yeah, to me, this makes this much more complex, thinking about that additional possibility.
I have a question. There's also a big picture. To me, it's like when Richard Semon was, or whatever his name was describing an engram, it's kind of like very theoretical. And this is really trying to make it into a paradigm that they can study. But I wonder if the whole idea of an engram is like just altered from what it was originally like meant to be. Like, I don't know. It's always like, is this really the engram that we're looking at? I don't know. That's what I'm always questioning when I'm reading these papers and listening to you.
I'm not entirely sure. So, I mean, the original idea of an engram, as Semon put it, is like, there's 4 components to it. I'm not going to remember exactly. It's some sort of substrate that changes specific to memory experience, that tracks the actual experience that was had, that can lie dormant for extended durations and then is reactivated upon behavioral recall. I think in all these experiments we're looking at, I'm not sure people ever mean to claim that like this particular set of cells in this particular, small region of the brain is the full engram for a particular memory. In that there are experiments where they show if you look at like engram recall and you use a like more whole brain sort of imaging technique, you can see activity across broad regions of the brain. And like all the information encoding a specific memory probably comes from a much larger cell, almost certainly comes from a much larger cell population than is seen in these experiments. Maybe with the exception of like, I don't know if you could see like all of the zebrafinch's HVC and RA, maybe that the full engram would be in there and not in other brain regions. But yeah, I'm not 100% sure what your question is getting at, but Ken?
So I'm confused as well. Let me try to. This is probably wrong, but let me try to simplify this and tell me where this is wrong. Okay, so so the way that I have been looking at this is CA1 is it is going to be representing where the animal is. And even if the animal doesn't know where it is, it's going to take the best guess. So it's like there's a set of representations that it can make, and that set of representations, even if it has very little to go on, it's going to default to one of those. Okay, and so there is in the linked, quote unquote. I think the problem is I don't know what the hell linking means, right at the front. It's like, why would you want to link these two memories? And what I'm saying is that in context B, where it actually got shocked like 3 or 4 times, okay, it's got a good solid CA1 representation of that, okay, and so that's going to go out. But in the quote unquote linked, it's got a good solid representation of A because it's been in A, and it's going to output that. And in the non-linked, the only thing that's not linking it is that it doesn't know where it is. And so if you don't know where you are, CA1 is going to take the best guess that it can. Okay. And the best guess might be, hell, I got shocked. So I think maybe I'm in B again. Maybe I'm in B again. Who knows, right? You know, CA1 is going to take a guess, right? And so what BA is going to either, and this gets back to our previous ones, BA is either going to light up an existing engram and say, hey, I recognize that. I got shocked here before. I do not need to form a new memory because I've got an input going. So, bam, I'm going to strengthen those synapses. Or it's going to say, hell, I've never been here before and I'm getting shocked. I'm going to take a new set of cells that is going to become a new memory engram. Okay? And in the linked case, it is going to take this is where this breaks down. This is how I've been thinking of it, but in that case, I would think that the linked would form a new set of engram cells in BA.
But they show that there is overlap between the engram cells for B and the engram cells that are active during the A experience, like the A prime experience later. So it's not it might be a partially new, but it's not a fully new set of cells at that stage in the linked paradigm.
Yeah. And those are in BA, right? So these are cells in BA for B and for A, and then overlap between the two. Yeah.
Okay, but in any case, the idea would be that this is less about linking and more about the CA1 defaulting to representing this new thing to be like the old thing.
Yeah.
Yeah, I'm confused.
We're all still confused.
Okay. I think I'm gonna default to a MAR level thing here. It's like what is the I need the functional purpose of it before I start talking about the implementational level and the algorithmic level. It's like there's some reason for this linking to occur, and I didn't see this in the paper.
Yeah. Is there something missing here where maybe you actually received a hint, some kind of background information from Tomás, Ryan? I mean, Ken, I think did he send the paper to you, or where did we get it from?
Oh, no, I hadn't seen
This was not.
paper.
Okay, this was not a paper he sent us. Okay, so there's no like.
It was.
Here,
This wasn't one of the papers. It's one of the papers he nominated for the prizes last year. So he put it in. Sent
Okay,
us too.
so there might have been some additional information of why he thought this was particularly important.
I don't think there was from the email from memory. I think these were just two he submitted with no greater context.
Right.
It does beg the question. It's like this paper has an awful lot about connectivity, and very little actual connectivity. It's like this optogenetically activating this causes these cells to fire. Okay, that's connectivity, fine, but it's not really. It's not like a connectome has been gotten or an egrasp has been done.
Yeah. Cool. Yeah, I think that is it for the paper.